The only macromolecule that we need to finish with is it's the nucleic acid. All right, nucleic acid. So nucleic acid is very similar to proteins and carbohydrates in that they are long chains of polymers. Okay, long chains of polymers that are made up of individual building blocks. Okay, so by now you should understand how to connect monomers together, right? There are two types of nucleic acids in our body. We have DNA and we have RNA. In this picture, we can see the something called the nucleus of a cell, and then here are the chromosomes. Okay, so the chromosomes are actually not colored. Yeah.
I was just gonna ask, I know we ended on enzymes, but I don't think we did the concept.
No, we did not. Yeah. So here's the thing, right? Thank you for bringing that up. If you go to, because sometimes we're not gonna have time to do all the concept check questions. So if you want to review them on your own or look at the answers, you can there they are. Okay. So under the quiz folder, usually you'll be able to see them. Okay. Yeah. So these few questions we haven't done yet, right? Okay. You can try them and then you can look at the answers yourself. Okay. So these are chromosomes, and they are not colored, but whenever we look at things under the microscope, a lot of times we apply dyes and colors to it to help highlight certain features, and so we are able to, see them a little bit better, right? The picture got cut off here, but it shows all the chromosomes that we have in our body. Does anybody know how many chromosomes we have? I heard 36, 40, yeah, 46, okay? 46 chromosomes. 23 from mom and 23 from dad, right? And then it comes together and you get the 46 chromosome, okay? So for the nucleic acid, it is composed of the triplet again, the C, the H, and the O. And then we also have nitrogen as well as phosphorus. Okay, so that's the N and the P. The monomer, the building blocks for nucleic acid, is something called nucleotide. Okay, nucleotide. Now, I wrote this down for you last time, but let me just quickly help you see this thing one more time. So for carbohydrates, right, we have the monomers. Carbohydrates, the monomers are called monosaccharide, right? You should know that because you just did your quiz. And then for proteins, what's the monomer for protein? Amino acid, excellent. And there are 20 of them, right? Okay. And then today we're learning nucleic acid. And we have nucleotides. So 4 for DNA and 4 for RNA. I'll tell you what those are in a bit. Well, it's already on the slide. There are two main types of nucleic acid. DNA, which is probably the one you are familiar with. Okay? It stands for deoxyribonucleic acid. Okay? Whereas RNA is something that, you might have heard of it during the pandemic, right? If you receive a Pfizer vaccine or a Moderna vaccine, it's a technology that's called mRNA vaccine, right? So we actually have RNA in the body. They are used in various settings. We will learn all about them later on in the course. Okay, so these are the two types of nucleic acid. So what is a nucleotide? Right? A nucleotide has 3 parts to it. Right? I should probably write down here for you. Okay, a nucleotide has 3 parts. There is a phosphate. There is a sugar and a nitrogenous base. 3 parts to it. Now, some people get confused. They don't understand the difference between phosphate and what is this P stand for? Phosphorus, that's right. Right? Phosphorus. Okay. So phosphorus is an element, just P on its own. Right? You can see it on the periodic table. Phosphate, if you don't already know, you will learn it in chemistry. It is PO4 3 negative. So phosphate, it's a group of atoms together. Right? It's not just P, but it also has oxygen in it. Okay? So phosphorus is not the same as phosphate. In our nucleotide, we have 3 parts: the phosphate, the sugar, and the nitrogenous base. Okay? It shouldn't be a challenging concept because when we did amino acid, we drew it out and we saw the amino acid has different parts to it as well, right? There's a carbon in the middle, a hydrogen on the top. Remember that? All right. So the same thing here. We have 3 different parts. Now let's take a look at the nucleotide structure. On the left side, we have a nucleotide for RNA. And on the right side, we have nucleotide for DNA. Let me explain to you. They both have a phosphate group. You can see the PO4, P, and then 4 oxygen, and then PO4 here. And it's connected to a sugar. By now, you should have an idea that sugar are ending with OSE, right? Like glucose, fructose, lactose, they all end with OSE. Well, this is another kind of sugar called ribose. Okay, ribose is a pentagon, right? And it's a sugar that you will find in the nucleotide for RNA. DNA nucleotide also has a sugar in it, but the sugar is missing an oxygen over here. Right? Everything is the same, but over here, it's missing an oxygen. Otherwise, it looks exactly the same as the ribose. Right? So that's why the sugar is called deoxyribose. It's a ribose, but missing an oxygen. Okay? So that's why this is ribonucleic acid, and here is deoxyribonucleic acid. Okay? So they both have phosphate. The sugar will be different. And then we also have the nitrogenous base. Okay, you can guarantee I'm gonna ask you about the difference between DNA and RNA on the upcoming quiz. Okay, so please understand. Yes.
Sorry, just a question. Phosphate was PO3 or?
PO4, PO4, 3 negative. Yeah.
Oh, PO4 and then negative
There you go.
3.
Yeah, 3 negative.
Okay, that's right up.
Okay, so let's take a look at the nitrogenous base. Okay. It's called nitrogenous base because it contains nitrogen. That's all. Right. There are 4 versions of the nitrogenous base for RNA and for DNA. Okay? We need to understand this because we'll build upon it. Right? We are going to be talking about DNA for a very long time throughout the course. So for RNA, the 4 possible nitrogen base would be A, G, C, and U. Okay? A, G, C, and U. I'll tell you what they stand for in a bit. For DNA, it is A, G, C, same thing, right? But instead of U, we have T. Okay? So T stands for thymine, that's how you pronounce it. And then U stands for uracil. So other than the T and the U, A, G, C are the same. All right? Let's take a closer look at the nitrogenous base. Now, you don't have to draw the shapes. I hope that's obvious by now. And I'm not going to ask you to identify them either. but here is something that you need to know. We can classify the base into two categories. We either call them purines or pyrimidine. Purine, pyrimidine, depending on how many rings it has in its structure. If it has two rings, this is called ring, right? Like the rings in the steroids, right? I hope you guys got the steroid question, by the way. Steroid has how many rings? Four. So this one has two rings, right? If it has two rings, then it's called a purine. I see people asking what steroid question. It's okay, it might not be on your quiz, okay? Only some people have the steroid question. Anyway, so this is the purine because it has two rings, okay? And if it has one ring, then it's called a pyrimidine. Okay? So I need you to memorize which ones are purines and which ones are pyrimidine. And you might be freaking out because it sounds difficult, right? But fear not. Just remember this phrase. Pure as gold. Pure as gold. You can remember pure as gold, right? So purines are the A's and the G's. Everything else is going to be pyrimidine. Okay? Remember, we only have 5 choices, right? A, C, G, T, and U. So A and G are purines. Everything else is pyrimidine, okay? You got it? So what do they stand for? A is adenine, okay? That's how you say it, all right? G is guanine, cytosine, thymine, and uracil. Okay. So, it will take some time to remember what they stand for, but we'll be using it quite often. Eventually, you will remember. Any questions so far? All right. What's the purpose of knowing this thing? How does it relate to the DNA structure? Well, this is the DNA molecule, okay? The best way to visualize this is to think of a ladder. Okay? Think of a ladder. If you take a ladder and you twist it, right? Twist it, then you would get the DNA. Okay? It's called a double helix. A helix is a spiral, right? So the DNA is 2 spirals intertwined together. Right? Double helix. The sides of the ladder is made up of the sugar and the phosphate. Right? So sugar, phosphate, sugar, phosphate, sugar, phosphate, just all the way around. And the rocks that you're climbing on in the ladder, that's where the base are going to be. So in the ladder, sugar phosphate backbone, and then the rocks, like I said, are the ACTG. Because we are talking about DNA, there are no U, right? Only T. And I want you to study this diagram a little bit, right? You might notice P and A always goes together, right? And C and G always goes together, right? So that's called the base pairing rule. It's always going to be happening like this. If you see an A, you will see a T on the other side. If you see a C, you will see a G on the other side. Now work with me here, okay? Just try to recall the information. A over here. Is A purine or pyrimidine? Yeah, see, easy, right? If you remember the phrase. So this is a purine. I'm going to put it right here. This is purine. And how many rings does a purine have? 2 rings, right? If you have trouble remembering that, the way I remember it is purine has 2 syllables, so 2 rings. Thymine over here, is that pyrimidine or purine? And how many rings? Good. Now let's take a look at over here. What about the G? So 2 rings. And C? 1 ring, right? Okay. And if you check for all of them, you will notice a purine is always going to be paired with a pyrimidine. And that is important because that way it ensures the width of the DNA is consistent throughout the entire length. It's always going to be 2 rings plus 1 ring, right? Or it's going to be 1 ring plus 2 rings. So the width across is always going to be 3 rings wide, right? Why is that important? Well, imagine a ladder, right? That, sometimes it's like wider and sometimes it's really skinny, right? And then wider, right? Then this kind of ladder is unstable, right? Structurally, it's unstable. It's easy to unravel. It's easy to break down, right? So by ensuring the purine and the pyrimidine are going together, it will always be the same width. It will always be 3 rings across, okay? As opposed to, 4 rings, 3 rings, 2 rings. Do you understand? Okay. So that's why we have to understand the base pairing rule. Now let's take a closer look at the structure. When we describe DNA, we use 2 key adjectives, okay? We say DNA are complementary. Complementary means if you know one side, then you can find out the other side, okay? Knowing one side allows prediction of the other side, okay? Because of the base pairing rule, A always goes with T and C always goes with G, right? That's called complementary. And we're going to practice that in a bit. Anything unusual about the diagram here? Do you notice anything strange in the diagram? Yeah, it's upside down, right? Okay, one side is upside down compared to the other, right? That is the second property of DNA. It is anti-parallel. Okay, it's parallel. That means the distance between them will always be the same. We know that already, but it's running in opposite direction. The easiest way to understand this is to think of roads, okay, like the highway. Right? The northbound and the southbound, they are parallel, but the traffic is going in opposite direction, right? That's what anti-parallel means. Okay. If you have a hard time seeing this, you can imagine people lying down on the floor, okay, head to toe. Okay, and then on the other side, it would be also head to toe, but upside down, backwards. Oops. There we go. Anti-parallel, okay? So what's holding the 2 strands together? We have these little attraction, okay? Little tiny magnets attracting them. Do you remember that term that we always use? What's that? I heard it. Hydrogen bond. There you go. Okay. So the C's and the G's, they are attracted to each other by 3 hydrogen bonds. Okay. These are important information. Please write it down. And then over here, the A's and the T's, 2 hydrogen bonds. So which one is harder to separate? Is it harder to separate the C's and G's or harder to separate the A's and the T's? Yeah, because it's like having 3 nails together, right? As opposed to 2 holding it. So it's going to take more force, right? So this is very important information because in some cases we have a strand of DNA and we need to find a weak point so that we can unravel the DNA. Okay, we'll learn about this in lecture 9. But you probably had this done on you during the pandemic, right? How do you know if you have COVID or not? What kind of test do you do? Nowadays we just do it at home, right? I don't think they even handle those kits out anymore, right? Like the one that you collect a sample and then you drop it onto the tray, right? To look for the lines, yeah. But before that, right? What kind of test do they do? It's a 3-letter test I'm looking for. Okay, yeah, something like that. No, they have to collect your sample and then they have to send it to the lab and do it. What's the test called? You guys know? Start with a P. No, nobody could answer me either in my other class. No, no. PCR, does that ring a bell? Yeah, PCR, no. Yeah. They just took my sample. I don't know where they send them. Okay, I just care if I'm positive or negative, right? But it's a PCR test. Okay, PCR test. If you don't know what that is, that's okay. But to do a PCR test, you actually have to understand the temperature that's required to separate the DNA. And that depends on how much A's and T's and how much C's and G's are in the DNA sample. Okay, so more on that in lecture 9. All right. If I give you this question on a test quiz, how would you do it? Okay. Knowing one strand, how can you predict the other side? Well, it's quite simple. Right? A always pairs with T. So you just write down T. Okay. And G always pairs with. Yeah, so go on. You guys understand? We gotta work on the coherence, but I think, hopefully you guys understand, right? A always goes with T, and G always goes with C. Okay, how many marks do you think this question is worth? No, I'm more generous than that. Come on. Yeah, 2 marks. Okay, probably 2 marks. Okay, easy though, right? All right, we're gonna do some math, right? Okay, math in biology. Woohoo, right? Actually, there's a lot of math in the second half of the course. So you're gonna need a calculator. Okay, good luck. Yeah. So suppose this. You guys have class on Friday? Oh, every day?
No, except for Mondays.
Oh, except for Monday.
Yeah.
Interesting. Okay, so here we go. Suppose the cell has 40% adenine. Okay, 40% adenine. Okay, 40A. Find the percentage of everything else. Okay, so I forgot to mention in this question, it's DNA. Right? I mean, you kind of know because we are asking for thymine. So if you have 40% A, which of these will also be 40%? T, yeah, because A pairs with T, right? However many T's, A's you have, you're going to have the same number of T's. So that's going to be 40% as well. 40% A plus 40% T is going to make up 80% of all the nucleotides, right? So 100% is everything. If you subtract the 80%, you get 20%. And this 20% has to be equally divided between the C's and the G's, right? So 20 divided by 2 is 10%. So that's how you do it. Okay. We'll do one more. Let's say we have 35% C. You do it. And then figure out the rest. I'll take it up in a minute. Okay, so 35% C means is also what?
35%.
Oh, very good. And that's 70, right? That means we have 30% left. Got it?
Yeah.
That's all.
Yeah.
So the other kind of nucleic acid would be RNA. Right? The key difference between DNA and RNA we already kind of talked about. It's the kind of sugar they have, right? DNA has thymine, RNA has uracil. And another difference is that where DNA is double-stranded, yeah. Okay, do you understand the 70%? Yeah, so 100 minus 70 is 30, right? And then the 30 has to be equally divided for the remaining letters, right? So 30 divided by 2 is 15. DNA is double-stranded, okay? But RNA is single-stranded. Okay, just one side of it. The backbone is also sugar phosphate, but like I said, it's a different kind of sugar, right? Ribose as opposed to deoxyribose. There are 3 kinds of RNA. Don't worry about it. We'll learn them later. I just want to show you that there's mRNA, rRNA, and tRNA. We'll learn their function later on in lecture 7. Okay, and that's it for the lecture 2. There are some conceptual questions you can try later. I want to do this one with you though, okay? This is an example of like application questions. Right? Most of the questions you see today was just factual recollection, right? Like to recall information. But as we progress through the course, there will be some application as well, where you really have to apply the stuff that you've learned. And here is an example of that. Okay, why don't you just try it out first, and then we will take it out. Try again, please. With Amir, with Ruth. Anyone else? Yes, very good. Okay, so let's take this up. It says a chemical reaction occurs slowly at 37 degrees. And then you add this thing called molecule X and the reaction speed up. So let's pause there, right? What kind of substances, molecule, would speed up a chemical reaction? An enzyme, right? We learned that last class, right? Enzymes speed up chemical reaction by lowering the energy requirement, right? It's like having a scholarship. You don't have to pay as much, right? I told you. So then, which of the following is the monomer of molecule X? If we know molecule X is an enzyme, then we should know it is a protein. So basically what the question is asking you to do is find the monomers, the building blocks for protein. Right, building block for protein is amino acid, and you should recognize amino acid is this one. Okay, we drew it together. So you see this question is very layered, right? You have to go through several concepts in order to find the answer. Right? So don't worry, not all the questions are going to be like that on the test, but expect a few Sure. Yeah, that's a good question. This one here, we just learned today. We see the phosphate, the sugar, and the nitrogenous base. So this here is a nucleotide, right? This here is a fatty acid. This here is some kind of sugar. Okay, like I wouldn't expect you to know that's glucose, but because of the hexagon, you should know it's sugar.
Sorry, an enzyme look like.
Oh yeah, sure. That's a good question. Yeah, an enzyme, it's I mean they look different shape, but most of them it's like a globe. Okay, a globe with some slots, and then you can fit like different things into the slots for the reaction to happen.
like in the.
Oh, like you wouldn't be, yeah. So this is like a very small part of the enzyme. Like you would have to have multiple amino acids together, and then you form the. Remember those pipe cleaner that I showed you? Like the whole thing. Yeah. Okay, that's it for lecture 2. All right, we are not going home yet. Sorry, I'm just gonna go a few slides for lecture 3, and then we'll go. Okay. When's the next quiz, anyways? Let me just take a look. I can't keep track of all the schedule. Okay, so this is today. I will have 2 more classes with you. Yeah, okay. I see. Oh, and then we have the lab. We have the lab on the 6th. Yeah, okay. So we're gonna do a few slides here first before we go. Otherwise we might not be able to finish. Okay. I will turn on the quiz 2 review this weekend in case you want to start preparing for that. Okay. Hopefully you understand the benefit of watching the review video. Okay. Anyway, lecture 3, cells introduction. So this is the organization of living things, right? We spend a lot of time in lecture 2. Actually, we spent 3 classes on it. 3 classes talking about the atoms coming together to form molecules. And then we have the bigger molecules called macromolecules. So the next lesson, we're going to go up another level. We're going to be looking at these things called organelles, which are like mini structures inside a cell that helps them function. Okay? For the rest of the course, until lecture 9, we will be learning everything there is to learn for the cell. And then we will do tissue in lecture 10. Okay. And then next semester, we will do the organs and organ system. So how big is the cell? It's really hard to visualize it, right? Like, I mean, when we do the microscope lab, we are going to look at some cells together, right? I told you I have various organs in the lab that I'll show you. But, let's try to visualize the size a little bit, right? If you think about an adult, right, the height, we would measure in terms of meters, okay? It's usually 1.3, 1.5, 1.6 meters tall, right? Depending on the person. So we use meters. When we talk about an egg, right? How big is an egg? Like about this size, right? Like, so that would be like 2 to 3 centimeters, depending if you buy the extra large or the regular size, right? Okay. Something like an ostrich egg would be, as big as a 10 centimeter. Okay, so like about this big. If we keep on going down, cells is measured in micrometers. I introduced micrometers to you before. 1 meter has 1,000,000 micrometers. Right? So these are very, very small. And the organelles are even smaller. And then the proteins and lipids that we're talking about, they're so small, we have to use nanometers to describe them. Okay? 1 meter has 1,000,000,000 nanometers. Okay, 1,000,000,000. So don't worry about memorizing these things first yet. You will learn it in math. Okay. Later on in math, you will learn how to convert between various metric units. I'm sure you're looking forward to it. What is a cell? Okay, a cell is defined as the smallest unit that still retains the properties and the characteristics of life. You memorize the 7 characteristics of life for this quiz, right? All those characteristics are still going to be there in the cell. Right? They will still be able to reproduce. They are able to maintain homeostasis. But if you go any further, any smaller than a cell, then those characteristics are no longer there. Right? If we look at the structure inside the cell, for example, if we look at the organelles, they no longer have all the properties of living things. So the cell is as small as you can go and still call it a living thing. Cells look very different. Okay? We have some bacteria here that forms a cluster, and we call them staphylococcus. You might have heard of things like strep throat, right? So that's caused by these kind of ball-shaped bacteria sometimes. We have these things here. Do you know what these are, anybody? Yeah, neurons, right? So they're in the brain and they look very different than the bacteria, right? In fact, they're actually much bigger than the bacteria. This guy over here on the right side is it's I guess it's hard to tell what it is. It's something called amoeba. Yeah, have you guys anybody heard of amoeba before? I spelled it wrong. Amoeba. Yeah, so amoeba is it's not plant, it's not animal. It's a category called protists. Okay, don't worry about it. I just want to tell you. Protists includes a lot of these single cell organism that lives in fresh water, okay, in ponds, right? Do you guys know kelp? Kelp, yeah? No, kelp is like these really big looking like seaweed stuff. Right? You can Google it, kelp. That's another kind of protist. Okay. People think it's plant, but it's actually not. So the cells look very different. Okay. Even the cells within the same organism is going to look very different. Your brain cells, it's going to be completely different than the, say, the kidney cells. Okay. Compared to the liver cells. They all look very different. However, all cells, regardless of where you find them, they will all share three characteristics. Okay? They all have a cell membrane. The cell membrane is what separates the inside of the cell from the outside. And sometimes it's also called the plasma membrane. Okay, just think of this as like a Ziploc bag. And then you're gonna fill up the Ziploc bag with some water, and you're gonna close it. So the watery stuff inside is called the cytoplasm. So all cells have a membrane. It has some fluid inside, which we call cytoplasm. And in the cytoplasm, you will have some genetic material. Okay, genetic material. So this is like DNA, right? Okay, so regardless of what cells you are looking at, they will always have these three things. Okay, you have these, a bacterium would have them, a plant cell would have them. Okay, I'm just gonna finish up with the cell theory and then we will go. Yeah, probably like 5-10 minutes. So the cell theory has 4 parts to it. Okay. Number one, every living organism is made up of one or more cells. One or more cells. So this is another organism that's related to the amoeba. Okay. It's called a paramecium. Okay, we will see this guy come back a few times throughout the course. Right? It's just one cell big, right? One cell living in a pond. You won't even see it, right? Because they are so small, right? It's microscopic. That's why it's like sometimes swimming in the lake is a little bit, you know, for me anyways, it's a little bit gross because there are so many of these things you cannot see, and inevitably they're gonna go into your body, right? Okay. Most of them are harmless, okay, but sometimes you might have heard of amoeba making their ways into swimming pools, and it causes kids to become quite sick, right? So, anyways, that's paramecium. This is something called C. elegans. It's like a tiny worm. Scientists use them to do research, and it's made up of about 3,000 cells. Okay, they are very useful for proof of concepts and during the initial stage of like drug testing and so on and so forth. Okay, they even make these kind of worms. They alter them genetically. When you turn on when you turn on blue light, they go in clockwise, and if you turn on yellow light, they go counterclockwise. Right. So it's you might think, what's the point of that? That actually is related to research in things like Parkinson. Okay, if you can implant lights that could turn on one color or another color inside the brains of people who have Parkinson, then maybe you can help control the trauma, just like you can control the the movement of the worms.
Like they're not
Are these like living, you mean? Yeah, they are.
like
Yeah.
okay.
Yeah, they are real things, real living things. You can genetically modify them. Yeah, which we'll learn later on as well. Okay, so what about humans? How many cells do we have? Trillions, okay? Trillions of cells. Right. Obviously, you had less cells when you were younger, but now we're adult, we have a lot more cells. Second part of the cell theory. Even though the cell is part of your whole body, but they can actually live outside of your body. If we collect a little bit of cells from you, we can actually put them in these petri dish, and we can feed them with nutrients, okay? That's what the red solution is. And then you can put them in a 37-degree incubator, and they would be quite happy. Okay, they can live outside of you for some time. Okay, we're not talking about months. Okay, but like if you be very careful with them, depending on what kind of cells you're talking about, they could go up to weeks. Okay, and they're what's that? Oh, it's just nutrients. Okay, so like some sugar, some amino acid, right? So they are quite happy on their own, and they might even divide in this environment outside of your body. Okay, so the cells, even though they're part of a multicellular organism, individually they are capable of living on their own. Of course, you have to supply them with nutrients in the right condition. So nowadays you can buy what's called a cell line. Ordinary people would not buy, like I wouldn't buy cell lines, and I wouldn't do anything with them right at home. But people who do research, for example, they can buy these cell lines, and then he has appointments every time. So he has to leave. Everybody's staring at him like, he did something wrong. He told me already, okay? Don't worry. And if you need to go, then go. No hard feelings, okay? Anyway, where was that? Yeah, so you can buy these cell lines, okay? And one of them is called the HeLa cells, okay? HeLa cells look like this. Of course, they are colorless, but like I said, you can put colors on them to make them look nicer, right? Okay? HeLa cells are very popular in many different research areas. They are used to study things like polio vaccines, tuberculosis, even cancer treatment, okay? And they played a role in, coming up with the mRNA technology to make the vaccine, okay? So lots of people can use them for research. And it came from this lady, okay? Her name was Henrietta Lacks, okay? Henrietta Lacks, right here. I am blocking her name. Right here, Henrietta Lacks. So HeLa, that's where the name came from, HeLa cells. Okay, so she had cervical cancer in the 1950s, and at the time, the doctors slash scientists, a lot of times they do both, right? They collected a biopsy from her cancer. Okay, so you guys know what a biopsy is, right? Like a little sample, right? And then they grew it in a dish, right? So I told you, if you grow regular cells, even if you give them like the best conditions, they might not be able to survive very long, right? But because these are cancer cells, they were able to keep on growing and growing, okay? They grow and fill up a whole dish, and they were able to take some cells and put in a new dish, and then they will keep on growing and growing. And they are still growing today, right? That's why they can sell them, right? People who buy these HeLa cells for research purpose, ultimately, all of them originated from her biopsy, okay? And that's one of the characteristics of cancers, something we'll learn later. They can divide and divide and divide. They are essentially immortal, right? So, how much do you buy them for? Well, for a cheap price of $486, you can buy 20 microliter. Okay? How much is 20 microliter? You can't even see it. It comes in a little small vial and it's frozen. Usually it's frozen at about -80. I've done these things, that's why I know. Right? So and you want to be able to see it. But like when I was studying, yeah. So why is this so expensive? Well, that's because you only have to buy it once. Okay, you buy it once and then you start growing them, and you have your own batch, right? They just keep on growing in your own lab, and then when you have a whole dish, you can freeze them down, and then later, if you need it, you can just thaw them, right?
But what is that use it for? What's so special?
That's a good question. Yeah. So HeLa cell is not the only cell line. A lot of cell lines actually comes from cancer cell origin, right? And we have different cell lines for different purposes. Okay. HeLa cell is popular because they have been studying for so long that they know so much about it. So it's easier to manipulate. Okay. Now, if that sounds like exploitation, you are absolutely right. So the family, the estate, after the lady passed away, the family actually sued the company, right? Because they did not get her consent to basically commercialize her sales, right? And they're making so much money selling all these cell lines, right? You can read about it if you want. I'm not going to go into it, but there was some back and forth between the lawsuits, and ultimately the family settled with the company, and they got some money, because of this situation. Okay, two more things, and then we can go. Number 3 of cell theory: all living things come from division of pre-existing cell. No one knows where the first cell came from, okay? But we know in order to get more cells, you have to divide from a pre-existing cell. We have trillions of cells now, but at one point, we started with how many cells? One cell, right? The sperm and the egg coming together, that forms the first cell, and then eventually it became us, right? Okay. How do what things form?
Like you.
Oh, like the person? Yeah.
Like coming from one cell.
Yeah.
It turns into
We will learn that in lecture 10. Yeah.
Yeah.
We'll learn it. Yeah. Yeah. My daughter actually recently asked us during dinner out of nowhere. So yeah, how do you form a baby? And then my wife, stared at me. It's like, you take the question. And then I was like, so daddy's gonna give mommy some DNA, and then you're gonna get it. Because she watched Magic School Bus, right? So she knows what's DNA. And she was like, oh, okay, I see. And I was, and a couple minutes later, she's like, so how do you give her the DNA? And then I was like, maybe we'll discuss this later. Anyways, kids are stressful. All right, last one, number 4. All right. Cells contain hereditary material which are passed on during cell division, right? So every time the cell divides, every time it makes copies of itself, it has to copy all 46 of the chromosome as well. So every cell that comes from the division is a faithful copy of the original, okay? And as you will learn later, also in the course, if it's not the same copy, if you make some mistakes when you're copying the cells, then it leads to mutation, right? And these mutations sometimes can cause cancers. Okay? And that is all for today, right? I'll see you guys next week. Thank you.